Hello guys, this is the second portion of chapter 9 and we should be able to finish this one today it's not going to, hopefully it's not going to take too long, maybe 45 minutes. I should be able to finish it up. Okay, what happens so far, we already talked about this a little bit, that what happens when pyruvate is made. You take a glucose molecule which has 6 carbon, it breaks down to 3 carbon molecule, which is called pyruvate. You remember this? Okay, so, and then you started out, I'm going backward a little bit. The cell invests in 2 ATPs, and at the end, when it's all over, then you generate 4 ATP. Why is that? Because 2 molecules of 1,3-biphosphoglycerate becomes 3-phosphoglycerate. Again, these steps you should not worry about, but when you take biochemistry, you should worry about these things. They will ask you questions. So you make 2 ATP here and 2 ATP at that stage, okay? Eventually everything turned into pyruvate. And then, so 4 ATP already invested, the cell already invested 2 ATP, that's not much. Okay, that is really, really for a cell. And of course you make 2 NADH. Remember that, we are gonna come back to this. 2 NADH from conversion of glyceraldehyde 3-phosphate to 1,3-biphosphoglyscerate. Okay, now what happens when, when pyruvate is made? Citric acid cycle completes the energy yielding oxidation. The citric acid cycle, this one, it generates some NADH. You already made 2 NADH from glycolysis, but it makes a few more NADH, and those NADHs turn from in the electron transport chain, they make more ATP, which I'm gonna go over that right now. What happens, conversion of pyruvate to become oxidized completely, yeah, it needs oxygen, okay, where is it? Right here, it needs oxygen. So oxidation of the pyruvate to acetyl-CoA, it is before the citric acid cycle begins, then pyruvate must become acetyl-CoA. When it becomes acetyl-CoA, then which glycolysis links the glycolysis to Krebs cycle. The steps carries out by a multi-enzyme complex. It's then catalyzed by enzymes, and it's not that boy, if you take biochemistry or some what was the class I take? He spent like a couple of months, 1 month, not a couple, I'm exaggerating now. He spent a month or 2 just right here, lecturing on this, right? Yeah, but anyhow, the class is not biochemistry. Biochemistry, they cannot afford time-wise to talk about that. But anyhow, when glycolysis occurs in cytoplasm, pyruvate is shifted into mitochondria, and in mitochondria, pyruvate becomes acetyl-CoA. Right here is a pyruvate. It has 3 carbons. You remember we talked about that? And they made these 2 carbons red because these 2 carbons actually get into the citric acid cycle, or Krebs cycle. And then these are their transport protein, another function of proteins, which you've been studying. So what happens to this CO2, this CO2 becomes carbon dioxide, which you exhale. Right now you're exhaling carbon dioxide, okay? So that happens to that CO2. Then these 2 carbons right here, we call them acetate, is combined to this molecule CoA. This molecule is a huge molecule. At the end, it has a sulfur. I don't know why textbooks do not draw them. I used to have them in my old PowerPoints. I forgot to put them in here for you guys, but sorry about that. But coenzyme is a huge molecule this big. If I was going to draw it, it would be that big, the drawing. It combines to these 2 carbons, and then the molecule acetyl-CoA is formed. During the process, NAD becomes NADH, okay? So NAD becomes NADH, and as the cell is making pyruvate to acetyl-CoA. Where does that happen? It happens in mitochondria. Okay. So the citric acid cycle, that's what I'm going to talk about next. I have not talked about citric acid cycle. Krebs cycle, another name for it, Krebs cycle. Dr. Heinz Krebs, he won the Nobel Prize for discovering the Krebs cycle. Of course, he found out the nitrogen cycle. There was another thing he figured out. He should have won 2 Nobel Prizes, but he won 1 Nobel Prize. Anyhow, he contributed a lot to the field of cell biology. The cycle oxidizes organic fuel derived from pyruvate, generating 1 ATP, 3 NADH, and 1 FADH2. What am I talking about? That the Krebs cycle, which I'm going to talk about that right here next, but I'm telling you that 3 NADH, 3 NADH, you're right. 3 NADH comes out from Krebs cycle, okay? And then 1 GTP, and then 1 FADH, FADH2, okay? FADH2 is equal to 1 ATP, GTP is equal to 1 ATP, and then, of course, the NADHs, NADH, and NADH, they give you 3 ATP. So the Krebs cycle, just by itself, 3, and then it's going around once, because one glucose molecule, with one glucose molecule, the Krebs cycle goes around twice. Remember that. So you have 3 NADH times 2, so 6 NADH, 2 ATP, of course, 1 GTP, and 1 NADH it gives you a total of the NATP, 3 plus 4 is 5, it gives you 10 ATP. Okay, I hope you got high. I got 10 ATP from one glucose molecule. And in the morning, you eat breakfast, you eat thousands and thousands and thousands of glucose molecules. Here it is. So that was just the Krebs cycle, okay? He is writing NADH, 3 of them right here, 1 FADH2, and then there should be 1 GTP on the bottom. So, and then here is, let's go from top. Pyruvate oxidation, pyruvate from glycolysis becomes acetyl-CoA, NADH, and then CoA, of course, is needed. So you have, that's pretty much is the cycle. And then, of course, all of these carbons from here, the first one went to CO2, you exhale it. The other 2 eventually are going to be exhaled as CO2 as well. Okay, so all of the carbons that is in the glucose molecule you eat in the morning, the mashed potato you had for breakfast this morning, all of that glucose molecule in mashed potato you had for breakfast eventually are going to become carbon dioxide. Okay, so right here, the CoA and the acetyl-CoA is giving you some of the diagrams. So the citric acid cycle has 8 steps. Okay, remember glycolysis has 10 steps. Glycolysis, the citric acid has 8 steps. Maybe I go crazy and ask how many steps. You should know that. The acetyl-CoA, the acetyl group of acetyl-CoA joins the cycle combining with oxaloacetate. Very important. You should know that. I'll show you some pictures. Forming citrate. Okay, and the CoA is kicked off. CoA goes away. The next 7 steps decomposes the citrate back to oxaloacetate, making the process cycle. The NADH, FADH2 produced by the cycle, relaying the electrons to extend from food to electron transport chain. Right here, so let's talk about citric acid cycle. Right here, we already talked about this, already talked about that. So that's the next one, and then after that, I will talk about oxidative phosphorylation. Okay, right here, so let me go back, sorry. Now, what happens, you should know that the two carbons, these two carbons are combined with these 4 carbons. It gives you a citrate which has 6 carbon, okay? Citrate becomes isocitrate. Of course, there is an enzyme. They wrote down the name of the enzyme right here, and that's how I got the job at Delta College, because during my presentation, I mentioned the name of the enzymes, okay? But you don't have to know them for this class. Okay, when you take biochemistry, absolutely, you have to know those names. But anyhow, so these are the 2 carbons that Heinz Krebs made radioactive, and he traced them, and he saw them right here, okay? Then from isocitrate, conversion of isocitrate to alpha-ketoglutarate, you make 1-NADH right there, okay? And then one of the carbons, this carbon is kicked off as CO2. Alpha-ketoglutarate becomes succinyl-CoA. So again, you make another NADH, and that carbon, this carbon is exhaled, well, you exhale it eventually as carbon dioxide, okay? And these 2 carbons came from pyruvate, the glucose molecule, the acetyl-CoA. And then you make 1 GTP. GDP becomes GTP, that is equivalent to 1 ATP. These are equivalent to 3 ATP, 3 ATP, okay? And then succinyl-CoA becomes succinate. Succinate becomes succinate becomes fumarate. You make, in that process, FADH2 is made. FAD becomes FADH2, and fumarate, oh, sorry, why do I do that? Fumarate becomes malate, and malate becomes oxaloacetate. And another NADH is made. So you made, in the cycle, you made 3 NADH 1 time. The cycle goes on 1 time. But remember, the glucose molecule you ate this morning, it makes 2 pyruvate. 1 glucose makes 2 pyruvate. So the cycle goes around twice. One cycle, the cycle goes around once, 1 FADH2 and 1 GTP. So the total, the cycle goes around twice. So that would be 6 NADH. That would be 2 FADH2, 1 glucose molecule, and then 2 GTP. Okay. Right here, he's going over the steps one by one. I don't expect you to know what becomes what, but I might ask you which of the following is an intermediate. That's the word they use textbooks. Intermediates of Krebs cycle. Okay, the intermediate Krebs cycle that I mentioned starts with citrate, isocitrate, alpha-ketoglutarate, fumarate, malate, and oxaloacetate. Anyhow, you should recognize it in a multiple choice question. Following glycolysis and the citric acid cycle, NAD and FADH accounts for most of the energy extended from food. That's right. Very important sentence. These 2 electron carriers, the FADH2 and NADH electrons to electron transport chain with power of ATP synthesized via oxidative phosphorylation. The electron transport chain is in the inner membrane cristae of the mitochondria. Most of the chain's components are proteins, which exist in multi-protein complexes. Yeah, the alternative we do is oxidative. Anyhow, electrons drop. Okay, let's talk about this. I talked about this, 10 steps. There's just 1 step right here, 8 steps right here. Let's talk about this guy, which gives most of the ATP. Out of these 4, multiple choice question, out of these 4, this guy gives the most of ATP to you guys. Okay, electron transport from NAD or FADH2 to electron transport, yes. By cytochrome system, yes, I will talk about that a little bit. You're not taking a biochemistry or another class, so I don't want to get into cytochromes too much. What is chemoosmosis? Very important concept. You should know that. What is chemoosmosis? You know the term osmosis. We talked about it. In this case, chemoosmosis is where the hydrogen ions go from high concentration to low concentration. Let's look at it. Electron transport in electron transport chain causes proteins to pump hydrogens from mitochondrial matrix into intermediate space, which I will talk about that. Hydrogen ions then move back across the membrane, passing through the protein complex called ATP synthase. That class I took, I guess it was, anyhow, he spent another 5 weeks, another 2 weeks on just ATP synthase. Oh my God. ATP synthase is just named enzyme, right? Remember that? Enzyme, ace at the end of the word, enzyme. ATP synthase uses the exergonic flow of hydrogen ions to drive phosphorylation of ATP. This is a chemoosmosis used right here. Okay. I wish we could, I think that this is the ATPase. He's showing you ATPase. I'll come back to this and I'll go over this a little bit more. But for now, let's focus what happens before ATPase. ATPase is the last step. And then that's your textbook should have not put this here. But anyhow, let's go. So what happens, the cytochromes you have throughout the electron transport chain, it's like a chain, look at this, it's chaining. It's a chain, go from I to Q, and then from Q to 3 to 4 to side of C and 4, and then finally your ATP synthase. Okay, that's a final product. So what happens, NAD that is generated from glycolysis, from conversion of pyruvate to acetyl-CoA, and also from Krebs cycle, they all go through these events. They go through the NADH becomes NAD, and when NADH becomes NAD, hydrogen gets out in here. Where is that? This is the cristae. I should write it down. The folds of the mitochondria, and out here, if you would, that would be the out 2 layers of membrane. You remember that? We talked about it. I'm gonna just draw lines. Okay, that would be your outer membrane. Okay, anyhow, you get the picture. So that is the outer membrane. Between the outer membrane and the cristae right here, okay, then these protein, the cytochromes complex, okay, they convert NAD to NADH to NAD, and hydrogen is pumped there. So there is accumulation of hydrogen ions between the outer membrane right here and inner membrane, the cristae. What happens then, all of that hydrogen gets up there. Of course, you need oxygen. This is the step that needs oxygen. I'm not going to elaborate on it too much. That's another deep discussion in people who take courses. But anyhow, then the accumulation of hydrogen here, right here, there is a whole bunch of hydrogen here. They go through ATP synthase, and ATP synthase pump them inside. Okay, and then with the help of that hydrogen, ADP plus phosphate is made into ATP. So scientists have sat down and worked very hard to figure out for one NADH, how many ATP is made? 3, roughly 2 point something. For one FADH2, how many ATP is made? 1. One ATP is made. Okay, so that's something you also should know. Okay, you should know one NADH makes 3 ATP, roughly 2 point something, one FADH2 makes one ATP, and one GTP is equal to one ATP, which you already talked about that. And this step right here is called chemoosmosis, right here. Hydrogen ions going from high concentration to low concentration. High concentration, right here, to low concentration. They're being pumped. There is a pump. And of course, ATP is being synthesized. If this is a pump that makes ATP, other pumps in the body that you studied so far, they do need ATP, but this is the pump that makes ATP. ATPase is a pump. Some textbook call it ATPase, some textbook call it ATP synthase. But anyhow, let's go. And then these steps are right here. It's called electron transport chain. I'm sorry, I wrote a lot. This is electron transport chain right here. Anyhow, so he's breaking down where oxygen is needed. What step? Step 4. And then you have the cytochrome Q, cytochromes, and so on and so forth. Cytochrome C. There's all of these protein molecules. They have names. You should not worry about it. Okay, here's an ATP synthase or ATPase. Another name for this is ATPase. For sometime during semester, I might refer to it as ATPase. So be aware that you know what I'm talking about. Okay, an accounting of ATP, production by cellular respiration. During cellular respiration, most energy flow is glucose to NAD, most of it, and electron transport chain, proton motive force, yes, and then even that's what he's talking about, proton motive force, that's ATPase, anyhow. About 34% of the energy in glucose molecule is transferred to ATP during cellular respiration, and making 32 ATPs. Eventually, it's 32 ATP. The rest is lost in heat. Okay, right here, he's breaking down how many ATPs synthesized for you from glycolysis. 2 of them, electron transport chain, citric acid. Sorry, this one it goes around twice, so you make 2 ATP, and then you make from oxidative phosphorylation, you make 26, 20 ATP, and you combine all of this. This is, of course, net. You remember I gave you those terms, net and gross. So the net is equal to about 30 to 32 ATP. Okay? So NADH is explaining where the NADHs are coming in this diagram, explaining where NADHs are coming from, FADH2 coming from, and of course the GTP on the bottom. You remember that? It is GTP. It says 2 ATP here, it's 2 GTP. Which cells use it just like ATP? If I go to Europe and bring euro here, easily I can go to gas stations, I wish, and give them euro and they give me gas, I wish. So the cells use it like that. Oh, what are these? Bypassing, okay, about 30 people, okay. What is fermentation? Let's talk about anaerobic respiration that the cells do. Most cellular respiration requires O2 to produce, and what the scientists believe fermentation is a primitive type of respiration. It was before the actual cellular respiration, aerobic respiration came. So anaerobic respiration was first, and then aerobic respiration. The cause glycolysis to anaerobic respiration, fermentation. Fermentation consists of glycolysis and the common types of alcohol fermentation. Right here, so what happens, they make alcohol same way as what we talked about, but there's no oxygen involved. So what happens, glucose go through glycolysis, becomes pyruvate, and pyruvate becomes acetaldehyde. And acetaldehyde, of course, you loses carbon dioxide, and then these 2 carbon becomes ethanol with the help of the NADH. This is the cycle. You should know that. Okay. What happens in lactic acid formation? I guess you exercised a lot sometime in your life. You have not been exercising, and then all of a sudden you decide, I'm going to go and run 50 miles. That's a lot. I'm going to run 3 miles, 4 miles. You have not been exercising. It's because of lactic acid accumulation. Your muscle cells cause the pain. So what happens? Glucose becomes pyruvate, and pyruvate becomes lactic acid. Lactic acid has 3 carbons also. Okay, pyruvate has 3 carbons also. You should know these things. You should know the number of the carbons in acetaldehyde, and ethanol eventually has 2 carbons, but lactate has lactate or lactic acid, same thing. 8 at the end of the word means acid. So pyruvic acid, lactic acid. Anytime you see 8 at the end of the word, it means acid. Okay, he is explaining the ethanol, and here he's talking lactate. Here is the diagram I pulled from another source for you. Liver, when you have not exercised, all of a sudden you go exercise, liver has a good amount of glucose for you guys as glycogen. Remember that? Okay, glycogen breaks down to glucose molecule. Glucose molecules they go to glycogen, and glycogen, the muscles also have glycogen. They break it down to glucose, and glucose becomes pyruvate. Remember that? Okay. Pyruvate becomes lactic acid, and lactic acid, before they go to liver, because there's a whole bunch of them accumulated around the muscle, right here, it causes the pain. Okay. So, but eventually in 2 or 3 days or a day, all of that lactic acid is pumped into the liver, and liver put them together as glucose molecule. That's what happens. An animation, let's see what it is.
All cells are able to synthesize ATP via the process of glycolysis. In many cells, if oxygen is not present, pyruvate is metabolized in a process called fermentation. By oxidizing the NADH produced in glycolysis, fermentation regenerates NAD+, which can take part in glycolysis once again to produce more ATP. The net energy gain in fermentation is 2 ATP molecules per molecule of glucose. Fermentation complements glycolysis and makes it possible for ATP to be continually produced in the absence of oxygen. There are 2 types of fermentation. Alcohol fermentation, which occurs in yeast, results in the production of ethanol and carbon dioxide. Lactic acid fermentation, which occurs in muscle, results in the production of lactate.
All right, we talked about it. And then the lactate cannot go pyruvate. There's accumulation of pyruvate, and pyruvate becomes lactate because there is not enough mitochondria. So when you exercise, you train your body, and you make myoglobin, and you make more mitochondria, and then when you run again, of course it comes to training, you will not get the pain. Obligate anaerobes, it means they are usually, we refer to bacteria that they cannot survive in the presence of oxygen. They're bacteria that they call them obligate anaerobes. Yeast and many bacteria are facultative anaerobes, meaning that they survive using either fermentation or cellular respiration. They're facultative. And then in facultative anaerobes, pyruvate is a fork in metabolic road to pathway. Okay. And evolution significance, I talked about this. So read it on your own, and I'm not going to talk about it. Okay, this is the very last concept in this chapter, very important. I would like to ask at least 4 or 5 questions, and during other times I would ask essay questions about this. So what happens, the food that we eat are made up of protein, right, carbohydrates, and fat. What happens to them inside of our cell? Right here, of course, the carbohydrates, as you know, they go through glycolysis, they become glyceraldehyde, and they become pyruvate, right? One thing that is missing in this diagram, pyruvate can also become glucose. You put 2 pyruvates together, they can become glucose, okay? Now, amino acid, some amino acids, they lose their ammonia group and they can become pyruvate. Fatty acids cannot. Look at this diagram. Fatty acids cannot become pyruvate. Some amino acids can become pyruvate. Glycerol also can become pyruvate. Glycerol becomes glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate can become pyruvate. Remember from before we talked about triglyceride? There is a glycerol molecule right here and 3 fatty acid attached to them. Remember that. Okay, so that's what I mean. Glycerol breaks from the fatty acids, from the 3 fatty acids, glycerol right here breaks and it can become pyruvate. Then what happens? Of course, the fatty acids can become acetyl-CoA. Okay. 2 carbons, for example, a fatty acid is this long chain of, I hate it when those lines appear. These lines, I'm sorry about that. Long chain of carbons, I don't know what to do. These 2 carbon break down and become acetyl-CoA. These 2 carbon break down and become acetyl-CoA. These 2 carbon, that's why 1 gram of fat has 9 calories. Of course, some amino acids can become acetyl-CoA. Okay. Then what happens? The acetyl-CoA can go through citric acid cycle and go through electron transport chain, give you ATP. If electron transport chain occur, it means citric acid must occur. Krebs cycle must occur in order for electron transport chain to occur. Citric acid Krebs cycle does not occur, electron transport chain does not occur. Okay, remember that. Very important. Third, feedback inhibition, most common mechanism of your textbook goes into that. Concentration begins to drop. That's true. If you do not exercise, you do not use the ATP, then all of these glucose molecules in your body become fat. All of the protein molecules in your body becomes fat. And that's what he's talking about. He's talking about feedback inhibition. Right here, that's a diagram that if there is no demand by cell, by organism for ATP, then all of that ATP becomes fat. So anyhow, this is my diagram I pulled from another source. I like this one because it shows you that glucose, pyruvate can become glucose, glucose can become pyruvate, pyruvate can become acetyl-CoA, acetyl-CoA cannot become pyruvate. Very important. Some amino acids can become pyruvate, and then pyruvate can become amino acids. It's a two-way street. And then some amino acids become acetyl-CoA. Acetyl-CoA cannot become amino acids. Acetyl-CoA cannot become pyruvate. And some other amino acids can become intermediates of Krebs cycle. Okay. Fatty acid, on the other hand, glycerol, we already talked about it, can become pyruvate. Fatty acids cannot become pyruvate. Fatty acids becomes 2 carbon fragments. These 2 carbon fragments become acetyl-CoA. And of course, if the cell does not need ATP and you don't eat any fat, okay? You eat carbohydrates and proteins, what happens? And you know ATP is needed, so all of these acetyl-CoAs turn into fat. All of these turn into fat. Okay, so that's what happens. Anyhow, that's the end of the chapter. I hope it made sense. And then if you have any question, please let me know.